Synthetic violacin, the power of designer pharmaceuticals in the real world. Present-day disease therapy has not been able to administer strong pharmaceutical agents without any detrimental impacts, including resistance and side effects. Especially in cancer cells, current medications for cancer, such as chemotherapy, have not been able to deliver strong restorative outcomes due to their non-targeted drug delivery. Therefore, a complex therapeutic strategy is required to fit the complex molecular nature of cancer. So how can we solve this? Violacein is a natural purple compound released by the bacteria Chromobacterium violaceum. Has been shown to have different anti-cancer, antibacterial, anti-tumoral, antiviral, and antioxidant properties, allowing the compound to be potentially used in a variety of types of diseases and drug delivery platforms. A huge problem is that commercially available violacein costs up to $395,000 per gram due to C. violacin's delayed production of the compound. The bacteria is pathogenic, so therefore violacin must be produced by a host which is easy to acquire and is a high production at a low cost. So to actually solve these problems, the bacterial strains that I primarily used as model hosts to produce the compound were E. coli TG1 and DH5-alpha, which would theoretically increase yield and reduce the price as they are fast-growing and non-pathogenic, critical for its industrial use. So this led me to my two main objectives. First, to extract functional DNA material from C. violacin, containing the genes to produce violacin, insert them into E. coli TG1 DH5-alpha and S-type Pharmarium and examine the violacin yield and quantify the price of the compound. My second was to examine if my inexpensively made compound exhibited any anti-proliferative properties on cancer cells. Also, if S-type Pharmarium was able to produce the compound, I would run simulations to verify the potential of a drug delivery platform. So to start, I split up my experiment into three phases, which the first was to produce violacin in a new host through transformation. So after production of the violacin was quantified, E. coli TG1 showed a 4.8-fold of violacin yield increase when compared to C. violacin. On the other hand, E. coli DH5-alpha showed no production of the compound at all, making E. coli TG1 the optimal strain and reducing the compound's price by over $300 per milligram when price of manpower, materials, and purification were calculated. Also, the S-type Pharmarium strain demonstrated production on demand, meaning it has the potential as a drug delivery strain, which I'll get into later on. So after purification of violacin was quantified, E. coli TG1, it was demonstrated using thin layer chromatography. It was demonstrated that standard violacin extracted from C. violacin possessed the same retention factor or polarity as violacin extracted from E. coli TG1 and the S-type Pharmarium strain, indicating that transformed plasmids did not get altered and both are the same substance. So after phase 2 of our experiment, so testing the cell viability of cancer cell lines, it was found that violacin is capable of causing cell death in HEY as shown through my dose response, where the x-axis represents the concentration of the drug, and the y-axis represents the normalized absorbance or percentage of survival. Although results show violacin being less potent than doxorubicin and cisplatin, you must also take into account violacin as a biologic with the potential to exhibit less side effects. Although in my previous slides I explained how violacin was less potent, my next graph shows violacin under hypoxic conditions, and it was shown that violacin is more potent compared to normoxic conditions, with a 6-fold decrease in hypoxic IC50 value. Of note, studies have shown chemotherapeutics are less effective in combating hypoxic cells or quiescent cells, making them mostly palliative, thus giving violacin a potential in killing chemo-resistant cells. So after examining violacin's antiproliferative activity on colon epithelial cells, it was found that inhibitory concentration that lowers cell viability by at least 50% on our cancer cell lines show toxicity to our normal cells, which chemotherapeutics have not been able to do yet. So to confirm cell death, microscopic analysis displayed that cells start to change in appearance at 100 micromolar, and at 1000 micromolar, cells are fewer in number, have a rounder morphology, and are detached from the plate. So using the power of synthetic biology, we've successfully bioengineered a novel strain of E. coli and Salmonella that can produce violacin on demand at a significantly faster rate than its original producer, making violacin a significantly cheaper substance. So in fact, the primary strain of S-type Pharmarium transformed to produce the compound may suggest the potential for a targeted drug delivery system of the violacin-producing strain to target a local tumor environment, potentially through an intravenous injection followed by tumor accumulation, drug delivery, immune activation, and then the bacteria can be killed off through the use of antibiotics. So at the moment, violacin is limited for researchers to study, but by making the compound significantly cheap It's cheaper. We have allowed more researchers to be able to test our compound and expand on my research, which has been shown promising results as a chemotherapeutic from testing its apoptotic necrotic activity, its impact on normal cells, its impact on other chemotherapeutics, its impact on cells with and without the p53 gene, the purity of the compound, its impact in hypoxic regions where chemotherapeutics are less effective in and more expensive than our new compound. And finally, in our 2D tumor model, where the growth rate of the bacteria produced enough drug to reduce the tumor faster than it grew.